Okay, so Watson and Crick have now figured out the structure of DNA. We know it's a double helix composed of anti-parallel strands that are running in, like I said, anti-parallel directions. And we can see here an example of that. Here's the 5' end and the 3' end. Here's the other strand over here. And we can see the bases here in the middle. Here's A, and there's a hydrogen bond to T, and C, and a hydrogen bond to G, so on and so forth. Now, what Watson and Crick figured out is that the structure of DNA kind of gives you a clue to how it's replicated. DNA is the only molecule that kind of directs its own replication. And it does that by the fact that it has this double strand that's covalently bonded together. So what happens is DNA separates itself in half, or it's separated by enzymes anyway, but it gets separated in half. The two halves come apart. Then raw materials are brought in. So for example, if you had a nucleotide T, it would naturally pair to A. And if you had a G, it would naturally pair to this. And if you had an A, it would naturally pair here. And if you had a C, it would pair there, right? So you can build a new strand of DNA using the old strand as a template. And this is known as semi-conservative replication. Your book will show you other types of replication. You should read about those. There were different hypotheses as the way DNA might copy itself. Semi-conservative means when you've made a brand new strand of DNA, so you separate this one in half, right? And then you build new strands, and then you end up with two new strands. So here's strand number one, here's the new double helix number two. Each one is semi-conservatively replicated, meaning it has half old DNA and half new DNA. So here's the new DNA on this one, and it's half old over here. So the strands separate, then this one is a template for building a new strand, and this one is a template for building a new strand. And you get two new strands of DNA that are half old and half new. Now, DNA replication, we now know, is always done from the 5' end to the 3' end of the DNA, meaning like in this example here where we're building a new strand, the 5' end is built first and the 3' end is added next. So it's built adding more to the 3' end of the growing molecule. Okay, so the growing molecule is always built by adding on to the 3' end. And the way this is accomplished is with the help of, first of all, you have the raw materials for DNA. So the food you eat and things like that brings in extra A, T, C, and G's because all of the food you eat has DNA in it, right? Because it used to be alive. So you have DNA in your food and you might make some of those components yourself as well. And then enzymes come in to help with the replication process. And we're going to talk about some of those enzymes and the process of replication in the next video. But before we do that, let's just remind ourselves of how DNA is going to replicate. And because DNA has what we call complementary base pairing, it means that a naked strand of DNA, so you split the helix, the naked strand is a template, and it allows it to act as a template to build a new strand of DNA because bases always complement one another. What does complement mean? It means if you have a T, you're always going to have an A stuck to it. If you have a G, you're always going to have a C stuck to it. So what I would like you to do, first of all, let's pretend this is the 5' end of this. That looks kind of bad, but there we go. 5' end and 3' end. That means the new strand of DNA that's kind of crooked, sorry about that. The new strand of DNA that we're going to build here, this will be the 3' end, and this will be the 5' end. So this new strand of DNA was built in this direction, okay? And so what would the bases be? So if it was paired to this C, what would that be? Well, that would be a G. What would be paired to this T? Well, that would be an A, right? You can draw it prettier than I am here, but you could draw this out. You could show me what the complementary bases would be that would be attached through hydrogen bonds to this template strand up here. I'll do one more for you. What would be here? That would be a G, right? So you can finish this one, okay? You can show me what those would be because I'm sure you'll have a test question like that. So in the next video, what we're going to do is we're going to talk a bit about how DNA replicates, and we're going to talk about some of those enzymes and the basic process that cells undergo to divide, and we'll do that in the next video.